In this video, we will explore protein post-translational modifications. These modifications regulate protein function, stability, and interactions. We will discuss various types of modifications and their importance in cellular processes and diseases. Let's start by looking at protein phosphorylation. Protein post-translational modifications refer to alterations that occur after a protein is synthesized during the process of translation. These modifications involve the addition or removal of chemical groups, which can include phosphorylation, glycosylation, acetylation, methylation, ubiquitination, sumoylation, netylation, palmitoylation, prenylation, ADP ribosylation, and hydroxylation. Each modification confers specific functional consequences to the protein, contributing to its regulation, localization, stability, and activity. Phosphorylation, a common post-translational modification, adds phosphate groups to proteins via kinases. This modification impacts cell processes by altering protein structure, activity, localization, and interactions. Signaling pathways are regulated through phosphorylation, which controls cellular responses. Glycosylation is a post-translational modification involving the attachment of sugar molecules, known as glycan, onto proteins. This modification influences protein folding, stability, and recognition by other molecules. N-linked glycosylation involves the attachment of glycan to the nitrogen atom of asparagine residues, while O-linked glycosylation occurs on serine or threonine residues. Glycosylation is essential for proper protein folding, trafficking, and cell-cell interactions. Changes in glycosylation patterns have been associated with some diseases, highlighting the significance of this modification in cellular homeostasis. Acetylation is a post-translational modification involving the addition of an acetyl group to lysine residues of proteins. This modification is catalyzed by enzymes called acetyltransferases. Acetylation plays a crucial role in histone modification, where it regulates chromatin structure and gene expression. Beyond histones, acetylation can also modify other proteins, influencing their stability, activity, and protein-protein interactions. Dysregulation of acetylation processes has been implicated in various diseases, including cancer and neurodegenerative disorders. Methylation is a post-translational modification involving the addition of a methyl group to specific amino acids, mainly lysine, arginine, and histidine residues, catalyzed by methyltransferase enzymes. Methylation can regulate protein-protein interactions, gene expression, and chromatin structure. For example, methylation of histones can affect gene transcription by influencing the accessibility of DNA to transcription factors. Methylation defects have been associated with diseases like cancer, cardiovascular disorders, and neurological disorders. Ubiquitination is a process in which a small protein called ubiquitin is attached to target proteins. This modification is carried out by a series of enzymes, including E1, E2, and E3. Ubiquitination is involved in protein degradation, signal transduction, and protein localization. Specific E3 ligases determine the specificity of this modification. Dysregulation of the ubiquitin proteasome system is implicated in diseases such as cancer, neurodegeneration, and immune disorders. Sumoylation is a post-translational modification involving the attachment of small ubiquitin-like modifier, SUMO, proteins to target proteins. Like ubiquitination, sumoylation is a reversible process and is catalyzed by specific enzymes. Sumoylation regulates protein-protein interactions, protein stability, and subcellular localization. It plays important roles in diverse cellular processes, including DNA repair, transcriptional regulation, and protein quality control. Dysregulation of sumoylation has been implicated in several diseases, including cancer and neurodegenerative disorders. Netylation is a post-translational modification that involves the attachment of the protein NET8 to target proteins in a process similar to ubiquitination. This modification modulates protein function and stability by altering protein-protein interactions and subcellular localization. Netylation has been shown to regulate processes such as cell cycle progression, DNA repair, and protein degradation. Dysregulation of netylation has been associated with diseases like cancer and cardiovascular disorders. Palmitoylation is a post-translational modification involving the attachment of a fatty acid, palmitate, to cysteine residues of proteins. This modification promotes protein membrane association and influences protein stability, trafficking, and signaling. Palmitoylation is dynamically regulated by specific enzymes, known as palmitoyl-acetyltransferases. Dysregulation of palmitoylation has been implicated in various diseases, including cancer, neurodegenerative disorders, and cardiovascular diseases. Prenylation is a post-translational modification involving the addition of a lipid molecule, such as a pharnyl or geranyl geranyl group, to target proteins. This modification promotes protein membrane association and influences protein-protein interactions and subcellular localization. Prenylation is catalyzed by prenyltransferase enzymes and plays crucial roles in signal transduction, protein trafficking, and cell proliferation. Dysregulation of prenylation has been linked to diseases like cancer and cardiovascular disorders. ADP ribosylation is a post-translational modification involving the attachment of ADP ribose to specific amino acid residues, such as arginine or cysteine. This modification is catalyzed by enzymes called ADP ribosyltransferases. ADP ribosylation regulates protein function, protein-protein interactions, and DNA repair processes. Dysregulation of ADP ribosylation has been implicated in diseases like cancer and autoimmune disorders. Hydroxylation, a crucial protein modification, adds hydroxyl groups to amino acids like proline or lysine. This maintains protein structure, stability, and interactions. Hydroxylases are enzymes that catalyze hydroxylation. Collagen, for example, relies on proline hydroxylation to maintain its stability. Dysregulation of hydroxylation is linked to diseases like cancer, cardiovascular disorders, and connective tissue disorders. In conclusion, protein post-translational modifications are critical regulatory mechanisms that influence protein function and cellular processes. They encompass various modifications, such as phosphorylation, glycosylation, acetylation, and ubiquitination, each with distinct functional consequences. Understanding these modifications allows us to unravel the intricate molecular mechanisms of cellular processes, aiding in the identification of therapeutic targets for various diseases.